Hello, my name is Ling. I will walk you through Module 6 of our synthetic biology class today. Before we start, please open the Google form attached to this module on the syllabus. In the module, we will look into commercial synbio products and regulations to make sure that these products are safe for human and environmental health. I hope that previous modules in the course have convinced you that there are endless possibilities as to what you can do with synthetic biology. Because of that, we cannot predict all and every single synbio products that will come into the market in the future. And so it is very challenging to create a safety standard that applies to all genetically engineered products. Not to mention that many synbio food products require long, large-scale, and expensive studies before they can definitively claim to be safe. Keeping in mind those two barriers to safety review of synthetic biology products, I want you to reflect on your experience with commercial synbio products and how you think they should be regulated, taking into account consideration for health and safety, as well as the feasibility of proving these products to be safe. And pause the video and answer the first question in the Google form, whether synthetic biology products should be assumed safe until proven harmful, or vice versa. Synbio products in the US are regulated under the Coordinated Framework for the Regulation of Biotechnology. Under this framework, new synbio products are subject to review by the US Department of Agriculture, USDA, the US Food and Drug Administration, FDA, or the Environmental Protection Agency, EPA. We will talk about FDA approval first. FDA approves GMO food products, including GMO plants. GMO stands for genetically modified organisms. Plenty of GMO plants have entered our food market since the 90s. For example, non-browning apples and non-bruising potatoes, plants that produce fish oils and rice with higher vitamin A, drought and disease-resistant corn and squash. Most of these plants, but not all of them, require that the manufacturer disclose that they are GMO plants on the packaging. On the other hand, FDA does not require GMO disclosure for imitation foods, such as egg whites and berries that are produced by not animal, but genetically modified microbes. So you will not know if the egg whites that you are buying is GMO or not, unless they are less nutritious than the real food counterpart. That's the only time when the manufacturer has to tell you that the food product is GMO. With that in mind, I want to know of your thoughts about the benefits and harms of GMO food products. FDA reviews applications for food product commercialization on a case-by-case basis. So the manufacturers may be required to conduct a pre-market safety review to prove that the product is safe, or they can go ahead and sell the products without any pre-market study. After the products enter the market, FDA is responsible for conducting inspection, testing, and enforcing safe manufacturing practices to detect and respond to any health and safety incidents. Traditionally, large companies have legal experts that coordinate with FDA in inspection efforts. However, with the appearance of DIY synbio tools, synthetic biology is becoming more accessible to home manufacturers, and there are concerns that FDA might not be able to keep all of the new GMO food products in check if they come from many delocalized manufacturers and are distributed among small communities. With that in mind, Do you think that FDA regulation should change and require GMO food manufacturers to conduct post-market safety surveillance instead of the FDA? Now that we've talked about plants, let's move on to animals. GMO animals undergo the same safety reviews as drugs used on animals, including animals intended for use as food, such as this GMO salmon that grows twice as fast as normal salmon. This means that the manufacturers must provide proof of safety and effectiveness of the genetic treatment before and after entering the market. Safety, of course, means that the animal is safe for human consumption, but it also means that the animal must not be susceptible to transmittable diseases. The animal must not show aggressive behaviors that can injure humans. As you can see, GMO animals are a case where potential risks to human and environmental health may not be detectable until after commercialization because they require a long time for the harm to manifest itself. That means that the FDA needs very intensive post-market surveillance to quickly respond to a rising risk. That being said, I want to know what other applications of GMO animals, aside from food, that you can think of. Now let's move on to the other category that the FDA regulates, drugs. While gene therapies have undergone clinical trials since the 90s, FDA only gives out the first gene therapy approval in 2017 and the second in 2019, which is quite late compared to China, Russia, and Europe, who approved of their first gene therapies in 2003, 2011, and 2013. That being said, many new gene therapies are being developed and undergoing clinical trials. Cellular and gene therapies are regulated as biologics, therapies derived from living sources like vaccines. Current successful gene therapies use viral vectors, which are viruses genetically modified not to cause diseases. We talked about one type in module 4, lentiviruses. These viruses are used to insert a gene sequence into the patient DNA. The new gene sequence take over the task of defective genes in the patient DNA. For example, sickle cell disease patients have a gene mutation that produces distorted red blood cells, which fail to transport oxygen. So a gene therapy is being developed that insert a functional red blood cell gene into patient stem cells. The new gene sequence can also provide new functions to patient cells. For example, to treat leukemia, patients themselves are given a gene sequence that produce anti-cancer antibodies. The stem cells are then injected back into the patient's body. These diseases are traditionally treated with stem cell transplant, where the patient is injected with stem cells from a healthy donor. The problem is that the patient's immune system might reject the donor cells and hurt the patient or render the treatment ineffective. With gene therapy, the patient's own cells are used, so no rejection happens. So please answer the next question about the technical challenges and ethical concerns that you know of around gene therapies. Let's move on to EPA-regulated GMOs. EPA oversees the commercialization of GMO pesticides. One type is pest-resistant crops, which are plants that have been genetically modified to produce their own pesticides. The other type is microbes, like bacteria, fungi, and viruses, which have been genetically modified to be toxic to pests. Both of these are exempt from EPA standards for maximum pesticide residue on food. So farmers can use any amount of these pesticides on food, and EPA would consider them safe for consumption. Before entering the market though, GMO pesticides undergo a safety review where the manufacturers have to submit information on the chemical properties and degradation times of any new protein. They have to prove that the pesticide is effective in killing the pest. They also have to prove that the pesticide is not harmful to animals, especially human and livestock, birds, fish, earthworms, and select insects like bees, which some of you may know are endangered. And the manufacturers have to prove that there's minimal risk of gene transfer to wild plants to minimize the impact on local ecosystem and habitat. After entering the market, the safety re-evaluation is required at least every 15 years. Despite efforts to reduce GMO pesticide impact on the environment, the pesticides have to interact with the pests to kill them. Many people are concerned that GMO pesticides will force these pests to evolve and become immune to pesticides. Do you think that that concern is valid? And if so, what can be done to avoid the risk? Another pest control method is to use GMO insects. The males are engineered to carry a gene that kills all female offspring and pass on to half of the male offspring. When released into the wild, these males mate with wild females and spread the gene, thereby reducing the insect population. This method was first done on mosquitoes to combat mosquito-borne diseases like malaria, Zika, dengue fever, and have seen great success in Brazil, where the mosquito population was reduced by 95%. It is now being tested on moths, caterpillar, and other agricultural pests. These GMO insects are regulated by USDA. The USDA requires a field study as proof of effectiveness and safety to human and livestock before large-scale release of these insects. However, there's a lot of controversy around how expensive these field studies must be to ensure that these GMO insects do not disrupt local ecosystems. Because even though these insects might be pests to our crops, they can be important food source to other animals, and there's always the risk of passing on the genes to other species. The Coordinated Framework for the Regulation of Biotechnology states that its goal is to provide safety without inhibiting innovation regarding synbio products. Of course, the safety of workers, the researchers, and consumers are the most important thing in considering synbio products for commercialization. But redundant regulations can add costs and delays in the research and marketing process that discourage innovations and encourage manufacturers to exploit loopholes in the regulations. Take a look at the FDA response for approving COVID-19 testing kits, for example. The first diagnosis test came out so late because these companies had to go through long and cumbersome paperwork for the approval process that led to inadequate testing and delayed response to the pandemic. FDA tried to make up for this mistake by exempting approval requirements for antibody tests, but now there are too many antibody tests out in the market. Some of them have zero accuracy, and now people are faced with too many choices of antibodies and faulty antibody tests that give incorrect results. To finish this lesson, please fill out the last two questions in the attached Google form. Thank you, and until next time.